Hello, everyone, and welcome to today's webinar, Introduction to Digital SPR and its Use in Assessing the Binding Kinetics of DART. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Nicoya Life Sciences. To learn more, please visit nicoyalife.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I would now like to welcome our speakers, Dr. Mike Piazza, Director of Applications Development at Nicoya, and Dr. Vicki Goldenshtein, Post-doctoral Associate at Duke University. For complete biographies on our speakers, please visit that presenter tab at the top of your screen. We're going to be starting off with Mike. Mike, you may now begin your presentation.
Thank you so much for the lovely introduction. And thank you for everyone who has taken the time to join us today for this webinar. For today's presentation, I will start off by giving you a brief intro to SPR, followed by a background of Nicoya's story and why we do what we do. Then we'll introduce you to Alto and how it works, as well as its unique capabilities, followed by highlighting relevant applications to your research. We'll start by going through some basic SPR theory. Surface plasmon resonance is a label-free optical method of measuring the binding interaction between two partners. The diagram on the left shows the basic assay format used in SPR. This consists of a gold sensor surface that performs the detection to us, followed by the immobilization of one of the binding partners to the surface, which we refer to as the ligand, while the other binding partner is then free-flowing in solution, which is referred to as the analyte. The binding, which is measured in real time, is directly due to a change in optical mass. The property of our sensor that we are going to be monitoring is called an absorbance peak, as showcased on the right side here. The gold is going to absorb light, creating this absorbance peak. The center position of this peak is going to shift as binding events. In this case, the analyte binding to the ligand occur on the sensor surface. The peak position will then give us our sensor response, which we will collect and use to determine our binding kinetics. So why use SPR? SPR is a pretty versatile tool which allows various different data to be collected. The workhorse of the SPR platform is determining kinetics data, such as association and dissociation rate constants, or the on and off rates, as well as the equilibrium dissociation constant, which is a measure of the affinity of your binding interaction. SPR is also useful for quantitation of unknown samples from solution, as well as useful in antibody characterization for epitope mapping and binning, or to simply screen your panel of samples for yes or no binding. Some of the benefits of SPR include its label-free ability, the real-time data collection, and its low sample consumption. So I'll give you a background into Nicoya's journey. Our mission here has been to improve human life by helping scientists succeed. And we've been working on this mission since our CEO, Ryan Denemy, started the company in 2012. He was frustrated with the lack of accessible and affordable instruments for label-free analysis, and so spent 5 years developing a better solution called the OpenSPR. At the time, OpenSPR was the first benchtop instrument for SPR analysis, and since its launch, has democratized this technique and helped hundreds of scientists get access to this powerful data type to help accelerate their research. This then motivated us to develop Alto, the world's first digital SPR system. Our commitment to democratizing SPR has allowed us to raise $20 million in funding and established a strategic partnership with Agilent. We are a team focused on supporting our over 500 customers across 50 countries and constantly pushing the limits of technological innovation. This has resulted in over 200 patents filed on our products and has allowed our customers to publish in over 170 peer-reviewed journal articles, which spans all disease types and therapeutic approaches. We have been accomplishing this by helping scientists simplify their research with both our benchtop OpenSPR and digital SPR platforms. They'll take this time now to go more of an overview of our Alto platform. After having thousands of conversations with scientists, we realized that while SPR remained a critical technology in every lab, the existing solutions made scientists compromise on their workflows and projects. If you're familiar with SPR and BLI, these challenges will be all too familiar to you and really tie back to the traditional fluidics of these systems. As the world's only surface plasma and resonance system powered by digital microfluidics, Alto revolutionizes real-time interaction analysis by taking the complexity out of SPR and eliminating the need to compromise on quality and time. The Alto consists of the fluidics-free benchtop instrument, which has a touchscreen-enabled built-in PC. The instrument is super compact and does not require a connected PC, so you'll only need about 1.5 feet of benchtop space for it. The instrument contains all the electronics, optics, and temperature control elements for operation. We then took all of the complex traditional fluidics found in other SPR instruments and integrated them into a disposable cartridge, which contains the fluidics, the SPR sensors, and our sample and reagent wells, as illustrated on the right here. By using a technology called digital microfluidics, we were able to move discrete nanoliter volume of samples throughout the cartridge by the application of voltage to these individual electrodes. And finally, our cloud-based design and analysis software lets your data be accessible anywhere, so you can check in on previous results and design your experiments before you even enter the lab. So what does this all mean to you? For starters, you only now need 2 microliters of sample for full kinetics, which is about 100 times less than conventional instruments. This means you don't have to worry about wasting your samples that took you weeks or months to produce, and you can start to use SPR as early as possible in your discovery process. From these 2 microliters, because of the work we've done in the fluidic handling, you get amazing data with minimal hands-on time needed. Alto is also designed to save your most precious resource, which is your time and your team's time. With its automated onboard serial dilutions, ability to handle crude samples without purification, no need for degassing, cleaning, or labeling, and its cloud-based design and analysis, this will save you 70% of time compared to other solutions. And now, because all of these fluidics are in the cartridge, the instrument is very robust and reliable, meaning there's no downtime and no maintenance or service contracts. The simplicity in the hardware is critical for instruments that are as high throughput as Alto, because with 16 channels, there is a lot that could go wrong in a traditional instrument. This also means the instrument is very compact, needing less than 2 feet of space on your bench. Traditional instruments also have limited protocol flexibility because of the need for these fluidic pathways and hardwares that are fixed. With Alto, we can completely change the fluidic design with a simple software update, meaning the protocols can continue to expand and improve, and you don't need to go out and buy a new instrument. Now, while we've added some awesome features and capabilities with leveraging these cloud-based tools, the workflow is still going to be familiar. You will design your experiment in a simple 3-step process. Then you'll use the instrument's built-in PC to set up and run the experiment. And then lastly, you'll be able to analyze your experiment with the click of a button with automated fitting methods and models and the tools that you'll need to analyze your data. The Alto platform is available in various options. Our most popular is our Nico System Pro, which allows for the full suite of kinetic determination, quantitation, screening, and epitope mapping and binning of your samples. Now, for those that are only interested in basic kinetics, we have the Essentials package with the ability to expand on these modules with the different applications available. All of these options are available in a local mode use or a cloud-connected instrumentation. Now, taking a closer look at the Alto cartridge, we can see how the samples are moved. This is achieved by the application of voltage to a grid of electrodes. By turning electrodes on and off in sequence, we can move them throughout the cartridge, from the storage wells to the SPR sensors. Now, although Alto only needs 2 microliters of volume, it still produces very high-quality data, with the pump-free design ensuring 0 concentration dispersion between sample and buffer. There's also no sample evaporation, which allows you to always get accurate kinetics regardless of interaction times. And you can maximize reproducibility with automated dilution on cartridge. So taking a closer look at how these various aspects work on the cartridge, we can see in this example how the dispense feature works. We load our samples into these sample wells, and then the DMF will automatically dispense 350 nanoliter solutions. We can see these as these tiny colored droplets moving across the cartridge here. Next, we will transport these samples as well as the buffer to our mixing zone at the top of the cartridge. In this zone, the samples will undergo the automated dilution. As we can see in this example here, we can see the darkly colored drops losing intensity and color as they're mixed with buffer, performing the on-chip dilutions, saving you valuable time and accuracy in the lab. Lastly, we can now see how the measurement is done on the Alto. In this example here, we can see the droplets oscillating over the sensor surface. Each of our cartridges have 16 integrated sensors, which have our gold nanoparticles at the tip for detection. Here we can see this colored drop oscillating over one of the fibers, which allows us to acquire the data for our association phase, followed by a clear colored buffer drop to determine the dissociation phase data. This data is then collected and analyzed to determine our binding kinetics. On this slide here, I'll provide a summary of the features that are available with the Alto package. Each of our cartridges have 16 integrated channels utilizing digital microfluidics to move our droplets around. Our sample capacity has up to 48 individual analytes that can be analyzed per cartridge, with a total of 240 interactions due to the 5 serial dilutions that are produced for each cartridge. We only use 2 microliters per sample to get all of these kinetics due to our automated dilutions. We are also crude sample compatible with very minimal fluidic maintenance due to all the fluidics integrated into the cartridge. And we can produce data for an affinity range measurement from picomolar to millimole. The applications, again, that we are able to accomplish on here are kinetics or affinity screening, quantitation, and then epitope binning and mapping. We're compatible with all the biomolecules that you would want to use for your lab, whether it's proteins, antibodies, nucleic acids, viruses, both purified or in crude sample form. And we have various different surface chemistries available to allow you to get the work done that you need. I will now move on to application overview of our technology. SPR is the gold standard technique used throughout the drug discovery process at every stage. Our products are such a powerful platform for proteomics because they're extremely versatile. They play a critical role and have unique advantages at every step of the biologics discovery process. It is applicable to all platforms, as mentioned, including proteins and peptides, antibodies, multispecifics, cell and gene therapies, and oligo-based therapies, and more. Alto provides critical data throughout each stage of the drug discovery process, from target ID to bioprocess QC, and spans all therapeutic areas, including oncology, immunology, infectious disease, and more. This drug discovery process is complex, and each of these stages requires different types of information to ensure the best drugs are being selected for further development. And so we built Alto to be able to provide value at every step of this process. This includes optimized protocols for fast screening of large libraries, quantitation of active concentrations, highly accurate kinetics and affinities, and epitope binning. In the early stages of hit identification and screening, Alto enables scientists to find that needle in the haystack. Because Alto can directly screen from crude matrices using only a few microliters of samples, it can be used earlier in the process to select better hits before requiring scale-up. After you find your hit, you'll need to quantify them, which can also deal with a large dynamic range and excellent sensitivity to help scientists identify the cell lines that are producing the most antibodies. Once these best hits are identified, we can then rank the hits to determine those with the best binding characteristics for the drug mechanism of action, and continue the cycle through rounds of engineering and humanization to further improve their performance. Alto provides extremely accurate kinetics and affinity results across a wide range of binding profiles with minimal hands-on time. And then finally, it is also critical for scientists to understand where on their target their drugs are binding, which they can do on Alto through our epitope binning, which is critical for the optimization of candidates and establishing IP. Now we'll go through a few of these examples. This study here showcases how Alto can be used to determine high-quality kinetics for characterization of antibodies against a particular antigen. For each of these kinetic determination, only 20 nanograms of antibody were required. We were able to determine binding profiles that demonstrated strong affinity for, in this case, the hemagglutinin antigen, with KD values ranging from picomolar to nanomolar. On the platform, this can be determined whether using a single cycle or titration style assay format or a traditional multi-cycle kinetic format, with both a direct or a capture coupling protocol for that. We also showcase how Alto is able to directly screen samples from serum or in other crude matrices. In this example here, we had 48 samples containing either serum or antibodies in the serum, and these were screened against 2 different antigens to determine specificity. The analysis showed 100% accuracy in capture of the anti-mouse antibodies against the H5N1 hemagglutinin antigen, and also showcased 100% specificity to only the H5N1 antigen and not the H3N2 antigen, highlighting how Alto can be used to screen your antibody panels quickly. With these same samples, we were then able to characterize their epitope diversity. In this example, on the Alto platform, we're able to use our epitope binning module, which allows for a 16 by 16 epitope binning experiment, providing 256 pairwise binning interactions. The format used for this is a classical sandwich bin, where we immobilize our surface antibody in each of our 16 lanes, and then proceed to antigen captured, followed by pairwise binning of each of the solution antibodies. This then allows us to monitor in real time to show which antibodies are sandwiching pairs and which antibodies are blocking pairs. Our automated analysis software then produces a heat map which showcases our binders and not binders, allowing us to group these into bins, as well as these overlaid curves with both tabular and sensorgram view. The last application we'll highlight will be our quantitation module. We also showcased how we are able to quantitate the concentration of these antibodies in serum. In this particular experiment, we again used the same anti-hemagglutinin antibodies against the HA antigen. The Alto automatically produces a automatically diluted 10 concentration standard curve to allow for a calibration curve, which we then are able to assess our unknowns against to determine the unknown concentration. In this example, we've showed high precision with less than 10% intracartridge air for the unknown concentration determination. The quantitation module also allows for a large 3-order of magnitude limit of linearity to determine your unknown concentrations. So this highlights the different applications that can be used on the Alto platform. Now, we would like for some participation from you in the audience. If you could please answer the poll question that has been shown on your screen. We would like to know, how often do you use label-free biosensors in your experiments? Do you use it frequently, meaning either multiple times a week or months? Do you use it once every few months? Are you currently not using it, but planning to do so within the next year? Or do you currently not use it and are just here looking for information about label-free biosensors? Please take the next few seconds to finish that. Thank you for your time. We will now take a look at the results. And we can see that about 32% of you use it quite frequently, which is great to see. Hopefully you get some Alto in your hands. A few of you use it every few months, and a few also use it or don't use it but currently plan to in the next year. And about over half of you currently don't use it but are just looking for information. So we encourage you to visit us at nicoyalife.com to further expand your SPR uses. At this point here, I'd like to pass it over to Vicky, who can continue on with her portion of the webinar.
Thank you so much, Mike. I'm going to talk today about our use of Alto in our research and how we utilize it to assess the binding kinetics of DART. And I'm going to start with an introduction to DART. So it stands for Drugs Acutely Restricted by Tethering. A normal chemical drug does not distinguish between the different cell subtypes, such as cell subtypes in neurons. If they have the same receptor that the drug targets, it's going to target it the same way on both cells. And so what we design in our lab is this delivery of the drug by utilizing a covalent system containing halotac protein, or HTP, that covalently interacts with halotag ligand, which I'm going to call now analyte, or just a small molecule, because Nicoya sort of have a different labeling system here. And so it interacts with its small chemical molecule covalently, and by tethering the drug that we interested into delivering to a specific cell subtype, we can actually distinguish those cells. So for example, you can see the red cell expressing our halotac protein, HTP. When we expose it to the HTL with the tethered drug, it accumulates the drug on the surface. So the effective concentration of the drug felt by that particular cell all of a sudden shifts to the left, while the tethered drug actually loses a little bit of its potency and it shifts to the right. And so what it allows us to create is this therapeutic window when we can affect one cell but not the other. And so DART allows us to basically combine benefits of the pharmacology with the cell type specificity of the genetic techniques. It is a covalent capture of therapeutics, and it has modular design, which allows us to deliver any drug of interest. And then here I'm going to showcase you just an example of the two neuron co-cultures, one having the DART beacon and one does not. And so stops firing. And even if you increase the dose, it still does not affect the second cell. We put a substantial effort into optimization of this covalent capture in order to be able to introduce it into the live animals. And we did it through chemical desertification of HTLs. And so in here you see our first generation DART, where HTL is this long PEG linker entering the tunnel of the HTP, and at the end of the tunnel, a covalent capture occurs. And as you can see on the left of the screen, we tested on neurons the capture efficiency of this system. And now I'm showing you our next generation DART, where we introduced several different chemical compositions that allowed us to increase this affinity between the HTP and HTL. And so the way we measured it, we just incubated the neurons expressing HTP with HTL containing biotin, and then we followed this with streptavidin and some fluorescent marker. And as you can see, we were able to increase the affinity by around 50-fold. As our control, we have a HTP, or the protein where we mutated the sites that participate in the covalent capture, and that results in abolishing this covalent reaction with the analyte. And so this little point at 100 micromolar indicates basically the ambient reaction of the HTL with the cells, which even at 100 micromolar is not binding. And so all this testing on neurons is very time-consuming and expensive. And as we still continue our efforts in order to optimize the affinity even further, and we're moving into protein mutagenesis and exploration of variability between the HTPs, we need a better way or quicker way to test the affinities before we go into the cell cultures. And so we turned into the SPR measurements. However, we have several experimental constraints that we needed to address that are unique to our research. The first one is the covalent reaction between the ligand and the analyte that does not allow us basically to reuse the sensor. So how would we solve this? The second problem is that we have a small molecule. It's a chemical, the HTL, and it's around 1 kilodalton in size. So can we detect the binding events on a sensor with such a small molecule? And so the team at Nicoya, including Ryan, Rachel, and Hansu, they worked extensively with us to see if we can actually address this, and if we can actually successfully use Alto in order to accommodate our needs. And so the protocol we end up doing is immobilization where capture molecule. This is to address the covalent reaction and reuse of the sensor. So the immobilization via capture molecule means that on the actual sensor surface, we attach a nanobody. So this nanobody is against GFP, and we express our protein, the HTP, or the ligand in this case, conjugated, we express it with the GFP marker. And so now we can attach our protein to the surface of the sensor, and then we can remove and reuse the sensor at the point of the GFP connection, because that is non-covalent. So now to address the analyte size, yes, so we have our HTL that is conjugated to biotin, and that is a really small molecule, and we were not able to see that binding event. So we decided to conjugate it with the streptavidin to make it a bit larger and easier to detect, and that actually worked really nicely for us. Furthermore, we had to use the single cycle kinetics, where you consequentially incubate your ligand with the analyte with the increase in doses, and that is because it's a covalent capture. So once the protein reacts with the analyte, you can't reuse that protein, and that follows with the measurement of the K-off, which in our case is going to be a covalent capture, so we don't really care about that portion on the graph. And just to go over the sensor, you could see the color coding. So basically where we put in our antibodies, where we put our analytes, and in yellow you can see the well where we put our protein. And so there are 6 by 8 wells for the analyte, so you can utilize it for exploring different concentrations if you don't really sure what are the affinities to approximate those, or you can use them for just the repeating the just for the statistical purposes. And so you have 16 sensors and 8 lanes. So each lane has 2 sensors, and so one of them is your measurement sensor, and another one is used for sort of the reference. And so for our reference, we use our dead version of the HTP, which cannot bind to the analyte, and that is to see any non-specific binding events, or sort of subtract the background from our measurements. And of course, we have two different analytes, and so we can basically design our experiments on this cartridge the way we like. So we can use half of it for the first analyte and the second half for the second. It's actually very convenient because we can modify and adjust the experimental design per cartridge, and this is a very flexible system for us to do very unique experiments on this cartridge. And now I'm going to show you the actual functional analysis. So again, we're using single cycle kinetic surface capture, and we use the titration method where the surface bound HTP is presented with either first generation HTL or the second generation HTL sequentially with a threefold increased concentration. So that is built into the system, the threefold concentration, as Mike was showing you, that the dilution happened automatically. So for our LSPR capture measurements, we sort of had an idea that about our spread and affinities. And so we had to basically measure our sample at different concentration in order to combine them together because they had too wide of a spread. And so for our first version of DART, having the HTL 0.1 and our HTP, this is the actual measurement that we've got. And as you can see, it's pretty consistent. There's not too much error and variability in it. And then here you see the HTL 0.2 interaction with our ligand. And as you can see, it really reflects, and it's kind of measured correctly, I guess, in both Alto and our neuronal assay. So this is a good confirmation, first for us that our measurements are good, and second of all, kind of like a validation for Alto measurements as well. And finally, I would like to showcase you a data where we actually use DART in live animals. So in here, we are interrogating the Parkinson disease circuitry using DART. So we interrogate glutamatergic inputs into the defined cells, into the striatum, either D1 or D2 cells that are implicated in the circuitry of the Parkinson disease. And so we inject halotype protein that is indicated in red into the left striatum, and we also inject at some point the HTL32 indicated in the blue. And we also implant the cannula in the left medial forebrain bundle, and that is to then deliver the 6-OHTA, which ablates the dopaminergic afferents to the left striatum. So basically that creates a Parkinsonian model in mice. I'm going to show you a video where we start with a stable Parkinsonism in a mouse, and then deliver our DART, AMPA receptor antagonist, into D2 cells. So let's see. So here's the Parkinsonian mouse, and you can see that it does only the left terms, and that's because we ablated the left striatum dopaminergic afferents, and it does not really wide turns, really short turns, and with a lot of freezing. And this is the same mouse to the right with the treatment of DART, and as you can see, it explores the entire box. It can turn both left and right, and it has a decrease in the freezing during its motion. Okay, and here is the summary basically of the videos with different animals that you saw right now. So we first performed the surgery of the two cannula insertions, and then quantified the locomotion and the turning. And as you can see, it was still able to turn left and right, and it made a pretty wide turn diameter. So this is pre-lesion. Then we performed the lesion, which created the Parkinsonian mice model. And as you can see now, they turn to the left, and the diameter of the turns decreased substantially. We also quantified the freezing, and as you can see, it went from non-freezing to up to 30% freezing. After delivering our DART tethered to a drug, we see the decrease in the freezing, and also we see appearance of the right turns and the left ones, and we also can see that the diameter of the turns resembles the normal pre-lesion mouse. When we did the same manipulations with the D1 cells, you don't see the same effect. And finally, when you actually target both D1 and D2 cells, as the standard drugs do, you do see the decrease in freezing. This is in the red dots, and by the way, the blue dots are sort of a reversibility of the treatment. But you do not see this improvement in the turn movements. And so what that indicates to us basically is targeting fewer cells resulted in a higher efficacy of the treatment. And so with that, this is the acknowledgement of the Tadros lab, specifically for Mike Tadros, who designed all and conceptualized the DART, and Brenda Shields, who primarily worked on this problem. Thank you so much for your time, and we'll open up to questions that you might have.
Victoria, thank you for that excellent presentation. And Mike, you as well. Welcome back. It is now time for our live Q&A portion of the webinar. So to our audience, for those of you who have not submitted any questions yet, please do so now. Just type them into the Ask a Question box and click Submit, and we will answer as many of your questions as we have time for. So great. We have so many questions that have already come in. I'm just going to dive right in. Mike, Victoria, I'm going to ask them to both of you. So whoever wants to take it first, go ahead. I'm going to just start with this one right here. How does HaloTag target the cells of interest?
Yes, so HaloTag has two components, the HaloTag protein and the HaloTag analyte that is captured. And so you express HaloTag protein on the cell type of interest using standard viral approaches like cream mice and AAV viruses. You can actually express that protein and in the desired cell subtypes. And then when you deliver the ligand or the analyte in this case, it's going to be automatically captured and at the increased concentrations on the surface of those cells that you defined.
Thank you. Next question asks, how might DART evolve into a targeted therapy for humans?
Right. So DART currently serves as the research tool, an excellent research tool for understanding the brain circuitry and understanding the underlying causes for disease, laying out the important groundworks for the development of the therapeutic advancements. In our research, we're currently successful in achieving whole brain dosing delivery through cerebrospinal fluid, for example. However, it's an invasive approach, so we are aiming to achieve the safe cell-specific delivery throughout the entire brain without intracranial injection. So that is the current limitation of this approach.
Thank you. Now, what future directions is your lab exploring with DART technology?
So our ongoing research is obviously still lays in enhancing this capture efficiency. Um, but we are actively also developing a version of DART that enables simultaneous delivery of two different drugs into two different cell subtypes. Additionally, we're working towards creating a specialized DART system that is able to target drugs specifically to soma of the neuron or to the dendrites, because you sometimes have different connections at different locations of the cell. And in addition, we're also directing our efforts towards developing a DART system with rapid induced reversibility. Right now, it's a covalent capture. So this versatility and precision of the DART technology will enable a much wider range of application in neuroscience.
Thank you. Does the electrode of the SPR sensor have to be made out of gold, or would a carbon electrode also work?
Yeah, that's a good question. So the SPR sensors themselves are the fiber optics that are integrated into the cartridge. We use gold nanoparticles on there. That is one of the basic properties of the localized surface plasmon resonance that we employ. So for the Alto and OpenSPR specifically, they have to be the gold nanoparticles.
Thank you, Mike. And what is the detection sensitivity in terms of analyzing molecular weight?
Yeah, that's another good question. I can take that. Yeah, so the size of the biomolecules that we use would be dependent on a few factors. One would be the orientation as to how the assay is designed. So that being the ligand, which is the molecule that's immobilized on the surface, and the analyte. In a situation where the ligand is much larger than the analyte, then the signal will be smaller as we detect that analyte binding. For a minimum size of the analyte, we've had success with running like small peptides of like a kilodalton to 1.5 kilodalton. Anything below that, the resolution becomes kind of challenging. Now, in the opposite orientation, where if the ligand itself was of a low molecular weight, then that opens up the door to allowing us to measure, smaller molecules, because the analyte will be larger and we can detect a larger binding response. But in the analyte method, I would say about 1 to 1.5 kilodalton would be probably like the lower limit that I would want to work with.
Thank you. Our next question asks, how do you prevent sample loss inside a cartridge? A special surface coating?
I'll take that one also. Yeah. So the cartridge itself is loaded with a cartridge fluid. So it's a very, very viscous solution, which has very minimal evaporation. So over a 24-hour period, the evaporation is something on the order of about 50 to 100 microliters. And we load about 8 milliliters of that. So it's very minimal. The samples are loaded into this fluid, which makes evaporation of our actual samples negligible. So we can keep those droplets on there for days without seeing any measurable evaporation.
Thanks, Mike. Next question asks, is DART currently limited to AMPAR antagonists?
No, we are actually continuously expanding our capabilities of the DART HDL toolset. So at present, our toolkit contains both AMPAR and GABA receptor antagonists and agonists, as well as other compounds such as diazepam, for example. And so additionally, we have developed a lot of DARTs with fluorescent traces that we use for histological applications. So using this toolkit, we continue to research our Parkinson disease model, as well as exploring addiction, anxiety, catalepsy, and other areas of neurodegenerative disorders.
Thank you. Next question. Would the epitope characterization experiment that was just presented also work with surface immobilized aptamers?
I'll take that. Yeah. That another good question. So yeah, so the actual like module for the epitope binning allows for basically a 16 by 16 bin of any type of biomolecules that can be sandwiched together. Obviously, one of the prerequisites would be that they're either, if we're working with antibodies, monoclonal, the antigens would have to be monomers with only one binding site. Now, with that being said, the sandwiching pairs can be aptamers. They can be, as long as these aptamers can both bind to the same antigen, then we could immobilize one of them as the surface sample, add the antigen, or I guess its targeted molecule as the antigen, and then add the second aptamer as the solution, and then we can see which aptamer pairs have different epitopes on these antigens.
Thank you. Next question. Are there limitations on sample viscosity? How about ionic strength, either very high or very low ionic strength?
Good question. There are limitations on the surface tension of the samples that we load. So if the surface tension is too low, or also drastically high, too low would result in a failed dispense. Too high would probably cause mist-sized droplets to form. But like the range is quite large. For this, we do recommend supplementation of 0.1% tween in all of the samples. This provides the ideal amount of surface tension. Now, if working with, say, crude like lysates, in those situations, the supplementation of additional tween is usually not necessary. And the viscosity of those samples, we have no difficulty in moving. So,
thank you. And Mike, Victoria, this question, it has multiple parts. So I'm going to read the whole question and then let me know if you want me to repeat any part of it. Thank you very much for this nice webinar. What is the typical analysis throughput with Alto? For example, how many samples can be analyzed in 24 hours? And what is the typical hands-on time?
If you're okay with that, Vicky, I can take it.
And I can then follow up with what our experience was.
Sure. Yeah, absolutely. So the typical experiment runtime, or the amount of samples that we can run in the 24 hours, it varies, to say the least. The reason it varies is it depends on the times that we use for the association and dissociation phases. So in a situation where, for instance, we have really tight binders with slow dissociation, we're going to usually want to incorporate a long dissociation time to accurately measure that off rate. Now, using the default parameters in the system for designing your assay, if we were to use the quicker single cycle kinetic format, which is the titration method, where you're going low to high concentration of your 5 analytes without dissociation or regeneration in between, we can run 48 interactions. So that's 48 with the 5 binding concentrations in about a 6.5 hour time frame. So if you're using that method on one Alto in a 24-hour time frame, you could get about just under 200, 190 something samples. Typical hands-on time, so setup time, I mean, we have nice templates for loading your sample information, such as concentration and sample names. The design, it's pre-designed in the ecosystem software for you. So you just basically tell it how many samples you want to run, input this CSV file with your sample names and concentrations. And then most of the time would be spent with loading your samples onto the cartridge. So the cartridge has a very similar layout to a 96 well plate. So if you want to use the multi-channel pipette to load your samples, you're more than welcome to do that. But a typical sample loading would be on the order of 15, 20 minutes, depending on how experienced you are with it. So the total hands-on time to start a test would be somewhere from 20 to 30 minutes total.
Yeah, I concur. This is exactly how much it takes. And yes, for us, for the specific single cycle kinetics, it takes around 7 hours. So we usually put it in the evening and come back in the morning and it's all done. But it can run also in the morning, through the evening. So this is 2 runs if you need to.
Thank you both. Our next question, this one has multiple parts as well. Are the chips reusable after washing? And following up on that question, do you have chips with streptavidin or protein AG that do not have to covalently bind the analyte?
Yeah, I can jump on this one too. So the chips reusable. So the cartridges themselves are one-time use. Now, with that being said, we do have quite a bit of capacity to run multiple interactions on there. For that, we have the ability to do a regeneration step in between each interaction. So you can analyze multiple ligand analyte pairs on the same sensor in the same experiment. However, those have to be loaded onto the cartridge and into the design of the experiment when you set it up. For capturability, yes, we have various different either kits available or the ability to help you design your own capture surface. So we do have a streptavidin kit, we have a protein A kit, we have anti-His. We frequently work with various different anti-species FC captures. We have streptactin XT. So we would on the surface, we would create this either streptavidin surface or protein AG surface. And then we would be able to, in the case of streptavidin, capture your biotinylated ligand, and then analyze various different analytes against that ligand. Or in the case of protein AG, we could capture various different antibodies, regenerate them off, capture another one, and do that multiple times on the cartridge.
Very good. Thank you so much. This question has come in several times, so we'll dive right in, and we have time to squeeze in a few more after this before we wrap. What is the cost of the cartridge, and how many experiments can be run on one cartridge?
Yeah, that's a good question. So the cost of the cartridges currently are $90 per cartridge. Now, if you buy in bulk, there is discounts for bulk purchase. Again, the cartridges are single-time use, but on the cartridge, we can run up to 48 unique interactions. So, if you're in a core lab with various different users on there, you could dedicate a few lanes to one user's interaction and then a few other lanes to another user's interactions. We can have multiple different ligands and analytes that are analyzed concurrently on the cartridge.
Thank you, Mike. Okay, and we're going to wrap with this one final question. We have time for one more. How crude or impure can the sample be when it is loaded in Alto?
Yeah, another very good question. I'll jump in there again, Vicky. Sorry, I've been hogging the screen time here. So for the samples themselves, like I said, we've cracked cells, like lice cells, spun them down to get all of the precipitate out, and thrown them onto the cartridge and pulled out express protein from there. So very minimal, pure samples to fully purified samples. The cartridge, because there's no tubes that can be clogged or anything that needs any maintenance for cleaning after, we're able to very easily move around all different types of sample types.
Thank you both for this excellent Q&A session. And I do want to point out that we do have questions for our audience as well. So I'd like to bring your attention to some questions that we have for you in the bottom left corner of your screen. Please fill those out and help us improve our webinar program for you. So thanks for your participation. Now, I do want to thank our audience for joining us today and for their excellent questions. This webcast can be viewed on demand, and LabRoots will send you an email when it is available for replay. And we appreciate you sharing that with your colleagues and friends who may have missed today's live event. Thanks for joining us, everyone. Have a great day. Bye-bye.